All right. Let's start over again. Let's move on to the dark reactions. The dark reactions happen sort of simultaneously as the light reactions of photosynthesis. The dark reactions in your book are going to be referred to as the light independent reactions. This is because they don't directly need light, I suppose. There is no light involved directly in these chemical reactions, but they can't function without light because the dark reactions actually need all of the products that come from the light reactions. For instance, so here is, of course, the chloroplast, as we looked at before. So this whole thing is the chloroplast. It's inside of a leaf cell, right? A mesophyll cell inside of a leaf. The light reactions are happening inside there, right, in the thylakoids. Now we're going to be talking about the stroma, what happens in the stroma. That's where the dark reactions happen. These light reaction products are NADPH and ATP. So every one of these things is pouring out NADPH and ATP. All right? So in order to do the dark reactions, we're now going to enter what's called the stroma. So let's just say, I'm going to say I've got a thylakoid and a thylakoid and a thylakoid, right? There's big stacks of these thylakoids inside the chloroplast. This whole thing is the chloroplast. So let's just say this is the chloroplast membrane over here, right? These are thylakoids. The dark reactions are not about grabbing energy from sunlight because that's already been done. The dark reactions are all about taking that energy that was stored on NADPH and ATP into a more permanent fixture. And what we're trying to make is sugar, because sugar is super stable, far more stable than NADH or ATP. But remember, from inside the thylakoid, I'm getting NADPH, dot, dot, those are the two electrons, right? Two hydrogen electrons, and ATP. So each one of these thylakoids is spitting out NADPH and ATP. So on and on and on. So there's plenty of these high energy molecules that are being produced during the light reactions, doing the electron transport chain and the pumping and the so on and so forth. And now we're going to use these high energy molecules to build a sugar molecule because a sugar is so much more stable. These things are very unstable. You're not going to build a tree out of these two molecules. We need to build a tree or build a plant physically out of something that's stable and high energy, and that molecule is going to end up being sugar. All right, so step one of what's called the Calvin cycle. The Calvin cycle is sort of the technical term for all of the steps collectively in the dark reactions. All right, this area, by the way, is called the stroma. It is the fluid that's outside the thylakoids but inside the chloroplast. This is a true biochemical pathway. One chemical leads to another, and so on and so forth. Each one of the steps requires a special enzyme that does that one tiny little step, and it passes that molecule on to the next. All right? So we're just going to go through it, and then I'll try to explain the significance of each one of the steps. So here comes from the outside world, comes the source of carbon. Carbon dioxide. Carbon dioxide comes from the atmosphere in this case, and the carbon dioxide is going to be leaking into that plant passively in through the cell membrane and then through the the chloroplast membrane. So the CO2 is already in the stroma fluid, okay? That CO2 is going to get picked up by a very specific high energy sugar, not so stable sugar, called RUBP. RUBP is short for ribulose biphosphate. It is a 5-carbon. Each one of these red circles is a carbon. Okay, it's a 5-carbon sugar. What I'm not showing you is there's also OHs and there's H groups and double bonded. So this is a fully fledged carbohydrate, simple sugar. Okay. What I'm not showing you yet is that this thing is basically riding inside of a large enzyme. This purple blob is the enzyme that runs this first reaction. It is called rubisco. Rubisco is short for ribulose biphosphate, biphosphatase carboxylase, which means it's an enzyme. The purple blob is a protein enzyme that has the ability to carboxylate rubisco, meaning we're going to take another carbon and we're going to slap it on there. All right, so this is a super, super important step. This carbon dioxide gets picked up by rubisco as well. This carbon gets bonded to that 5-carbon sugar and gets spit out the other side as a 6-carbon sugar. So this sugar is now added 1 carbon. There's the new one. I'm going to add this one as a blue carbon so we can kind of follow where our new carbon came from. So here's our new carbon. Here's the old carbons. Here's all the old carbons. There's that brand new carbon. So now we've extended that sugar by 1 carbon. All right. This is a super important step. It's called carbon fixation. Carbon fixation is when carbons are taken from the outside environment, the atmosphere, fixed to an organic molecule, and now it's permanently attached to an organic molecule. So we've taken an inorganic carbon, added it to an organic molecule. All right. Next step, this 6-carbon sugar very quickly falls apart because it's very unstable now. This thing can't handle the fact that it's been messed with in this way, and so it splits in 2 right down the middle. Now we have 2 3-carbon sugars. We call them PGA. Phosphoglyceric acid is a fairly stable, high-energy sugar. The problem is that it's so stable that it won't do any further chemistry the way it is. And so PGA is kind of important. We use it for a lot of things ourselves metabolically. The plant right now can't do much with that because the plant's trying to make sugar, like a high-energy sugar called glucose, that is much more interesting to play with and to make things out of and to store and so on and so forth. So PGA is a good intermediate sugar, what we call an intermediate metabolite in this cycle. All right, now we need to add energy to this thing so that it can go on and do more chemistry. So think about where the energy for this process is going to come from. I've got a low energy, very stable 3-carbon sugar, no fun to play with, not going to do any more chemistry. Where can I get energy from? Well, over here, there is a ready source of energy that is ready to go. There is ATP pouring out of this thylakoid here. There is NADPH carrying high energy electrons around. Okay? So the first thing that happens is 2 of these ATPs show up. One there and one there. And they drop off. Let me pick another color for phosphates. They drop off phosphate here, a phosphate here, a phosphate here, and a phosphate here. Now we have a very excited molecule. Dropping off phosphates is called phosphorylation. When you take a high energy phosphate from ATP and drop it off onto any other organic molecule, that energizes that molecule and makes it a lot more likely to do biochemistry in the future, right? And so we're sort of just kicking this thing into action by throwing high energy phosphates at it. The next thing that happens is NADPH comes along, drops off a pair of high energy electrons there, right? High energy pair of electrons and the hydrogen there. High energy pair of electrons and a hydrogen there, and so on and so forth. So we're going to use up 4 NADPHs, 4 of those, and drop off those high energy pairs of electrons and hydrogens directly onto the PGA. Well, now we have a molecule called PGAL. PGAL is a high energy product of all of this energizing additions of molecules to this. So PGAL in your book is going to be called 3PG, 3-phosphoglyceric acid. And this molecule is a super important intermediate molecule because it can go and do a lot of other chemistry. The main thing we want it to do is if we take two of these and bind them together, remove the phosphates, we will automatically end up with glucose, which is what we're trying to make. 1, 2, 3, 4, 5, 6, right? One of those is our new one. One of those molecules is, one of these carbons is the new one that we brought in up here. That's the same carbon. These 5 red carbons are the ones we started with RUBP. This now can leave as a glucose molecule. The plant can take that glucose molecule and make it into bark or wood or another leaf or some cell membrane or cell structure someplace. This is how all plants make their sugar to build their structure and to store energy for next year. The problem is, right now, that we've used up the 5 carbons that make RUBP to make this sugar, so this process is going to end if I don't somehow manage to get RUBP back, all right? So imagine this thing goes around 6 times. I do this same process with 6 different RUBPs, 6 different rubiscos, so on and so forth, 6 different carbon dioxides. If I do this 6 times, I will be fixing 6 brand new carbons from the outside, which means I can take a glucose out, right? All of the other carbons from PGAL have to be returned, and this is another 3-step process. There's 3 more reactions here. All of these carbons, these 5, have to be returned to make more RUBP, because if I run out of RUBP, I can't fix carbon. If I don't have any of that RUBP, the whole thing will stop. So in other words, in order to make sugar, I need to do the Calvin cycle 6 full times, and then I have fixed enough carbons from the outside world, brand new carbons, that I can actually remove a glucose. But remember, as long as the sun is out, every plant or every cell that has chloroplasts in them, they're making thousands and thousands and thousands of sugars per second. And that's because this isn't just one cycle. There are thousands of Calvin cycles going on in every single chloroplast, and there are hundreds of chloroplasts per cell. And so there's always going to be a surplus of RUBP, and there's always going to be carbon in the atmosphere that you can pull in, turn into PGAL. That PGAL, as long as there's enough of them, you can combine 2 of those, turn them into glucose. That glucose that can then be used for the plant. Incidentally, this same glucose is the glucose that then goes and feeds you and me too. Everything we eat, doesn't matter if you're just eating pigs and cows, every single molecule in that pig or cow or salad came from this process, every single one of those things, all right? So that's essentially the Calvin cycle, and we'll finish up with another video later. We'll combine the 2 things together.